Alright, in this last video we're going to be talking about transcription start sites, and if you don't have the mRNA, this is pretty hard to really nail down sometimes. Of course, if you have the mRNA, the definition of a transcription start site is, well, where does the mRNA start? So if you know the sequence where the mRNA begins, you have the transcription start site. That is +1, as they say right here. That is the first letter. It should be, it might be an A for an AUG if there's no sequence in front of it. But sometimes the mRNA doesn't start at the start codon. There's a little bit of sequence in between there. In between the, say, -10, where it initiates, but where does it start actual copying, might not be at the start codon, it's somewhere in between. So the actual start site is often a guess unless you have experimental evidence in the laboratory of, oh, we took this RNA and we sequenced that RNA and this is what we got. And even then you might get differing answers. Sometimes it might start at one particular letter and then maybe the next time it starts at the next letter down, so you get a mix. So it's really not that important in the sense if you have experimental evidence, but you do need to know as a general rule what to expect, and you can go find it, of course, if you do have an mRNA. And we have the mRNAs for the lac operon, so you can just go look at it and see what the first letter is, find it, and say, oh, it starts at -7. All right, so for instance, that's not what I wanted. Like right here, we have our start codon, so you know it starts no later than -1. So it should or 0 at 1. It could start right here, but it's unlikely. It's probably going to start somewhere in this sequence here. It's probably going to start, assuming that this really is the TATA box right here, then it's going to start somewhere between here and here. And so the start, not the start codon, but the transcription start site, the TSS, is probably going to be in here. Now, there are some general rules that aren't real strict that that will give you a hint as to where it's going to start. And so we go through those rules and we can guess at it using our rules. We know essentially in the probably 20 base pair here, wherever, actually maybe only 10, depending on where it is. But if we actually think that it actually is at -10, then let's say -10 from our TATA, TATA. Let's go find the TATA and count 10 over. So let me show you how to do that, I guess. And we could say, well, if TATA is right here, right here where I have it, we count over from the beginning of it. All right, so 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. That could be our start, but it's usually, if you read the what I've written here, it transitions from AT to GC or vice versa. So let's go look and see if we can use that as a hint. Oops, not that one. Is there a region close to this where we transition to GC to AT? Yeah, it looks like it does. It transitions right about here. We get more AT here, more GC here. So my guess, according to that rule, would be the transcription start site is right here between the GG and the AA. And now let's see if we count backwards -10 and see if we don't end up right here. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, well, 11, 12, 13, 14. Close enough. So using our rules as we have kind of gone, my best guess for the transcription start site would be here. And so you just need to put that in, that the transcription start site would be right here. And I'm just going to mark that. I'll put the GA right there. It's a transition from high GC to high AT. And let's see, I'll just bold that. and leave that like that. And it's right there in the middle of the promoter. So the GA is right there, and TSS is bolded. All right, I'm going to put a note there. So GA, and I'll put that in parentheses. There we go. So I remember that. All right. So right there, our promoter. This is kind of what we're guessing at, and we sure that works. Now the thing is, if the RNA, there needs to be a Shine-Dalgarno. So if the Shine-Dalgarno needs to be there, remember it has to attach to the RNA. Is that enough? Well, this front piece could be enough. You might not need all of this for the Shine-Dalgarno. So maybe it is just the GA, but it could be one over. That adds some more G's to it, but actually kind of makes sense. So in other words, our transcription start site starts right at the Shine-Dalgarno, and that conserves space. So I kind of like that one a lot now that I see it. If this is our start site, then we start here. We've got a Shine-Dalgarno, and we're ready to go. We don't waste any space at the beginning of the sequence. So yeah, I like that argument that is indeed the transcription start site. Now let's read more about this. I have this paper here that I want to talk about. You can read this about how transcription start sites, and of course if you have the mRNA, you will know the transcription start site, because the beginning of the mRNA is what a transcription start site is. It's where the mRNA begins. So let's look at this paper though, and I just want to talk about it briefly. First, let's talk about what horizontal gene transfer is. You need to know that to understand what they're getting at in this paper. And horizontal gene transfer is just when basically DNA is transferred from one bacteria to another. In other words, there is no sexual reproduction in bacteria, but bacteria do have the ability to naturally uptake DNA in their environment, and they will incorporate it into their genome. Plasmids are an example of this. But sometimes they just take up DNA by accident when it's floating around. Other bacteria around them have lysed, they've died, and there's DNA floating around, and they will uptake it and they will incorporate it into their DNA. And that is, whenever this happens, either by plasmid or just by accident, we call that horizontal gene transfer. It's basically the transfer of DNA from one bacteria to another without the use of any intentional transfer, like I've forgotten the process, but one type of bacteria can create these spikes where they actually transmit DNA into they get infected by a plasmid, and then they make the spikes, and I've forgotten what that's called. But and then that allows them to basically bump into the other one and transmit the plasmids. But this case is a little different than that. This is just more or less accidental. And so using this, they're studying horizontal gene transfer, all right, horizontally acquired DNA in bacteria. And they're using this to figure out how that DNA gets incorporated, and they're studying the GC content and the AT content. And that has led them to some conclusions that GC-AT content is important for this transfer, this horizontal gene transfer, to take place. And so they study this, and they study all the GC, and so it's definitely bioinformatics, because they're really studying the GC and the AT content of the pieces that actually get transferred. In other words, if you have certain GC-AT content, you're more likely to be incorporated into the new organism. All right, so let's go down. Come on. How did it get so small? There we go. Oh, that's not good. Oh, there we go. That's better. Okay. So what they're showing is that the Sigma 70 actually has a stringency content. Even if the DNA is incorporated, they find that there are certain GC requirements for the Sigma 70 part of the RNA polymerase to actually initiate transcription. We just saw that with the requirements for GC and AT around the promoter. So this paper is actually goes into what those requirements in the new or what they're saying are. They figured out that the Sigma 70 uh, enzyme is actually requires these GC things, and it has a lot to do with. And I don't expect you to read all this, but the discussion is good. So here we have identified a mechanism to explain regulatory compatibility between bacteria and diverse genomic compositions. So, in other words, if you get a certain piece of DNA incorporated, it might not work in the new one because the Sigma 70 and this other species of bacteria, it has a slightly different Sigma 70. It's not going to be compatible with the GC content, and it's not going to be utilized. And so this is a way to maybe keep out foreign DNA by having your Sigma 70 being specialized for the GC content in your genome. And so different species, if you have a different Sigma 70, then your genes need to be have a different GC content than other species, and their DNA won't get incorporated into yours, that sort of thing. So that's what they're studying here, and the stringency required for Sigma 70 mediated transcription may vary across different bacteria species. So this is really what this whole paper is about. They're just saying that, hey, this GC content is important uh, for having DNA work, and this has lots of implications for what? If you're doing genetic engineering and you're working with K12, you need to know this because you may want to get a gene from another bacteria or maybe an artificial one you're creating. You may want it to work, and for some reason it's not. And it may be that you need to go through and add some GC content. So remember, there are codons that are used for a single amino acid. There are many codons. And if you need to increase the GC content for that gene to get it to work in this species, then you need to use the codons that will increase that GC content, and or lower it. You may need to lower it to get it to work in the organism. So we know that from previous video, I told you that we have roughly 50-50. It's 51 GC, I think, 51% GC. You need to get that as close to that percentage as possible for your gene, and especially get it right for the promoter region that we just looked at, for the Sigma 70 to work on and work efficiently in your new organism. You gotta set the codons, all right, correctly across your genes so that you get the right GC percentage to work in that organism. And so this is an important paper. This tells you how to successfully get your gene, when you're transferring it from one organism to another, to actually work well. All right, it might work, but you just don't get the throughput. It doesn't work as well. You're wanting it to produce a particular protein, and you got the gene in there, you got the plasmid in there, all right, and you transferred the sequence into the new organism, but hey, it just doesn't make as much as we thought it would, or it's making way more than we thought it would, and we don't want it to make that much. Well, how do you fix that? Well, what they're telling you here is that you need to adjust the GC content, and to do that, just switch to some codons that have more or less GC in them so that it will work better with your enzyme that is in your organism versus the organism that the gene originally came from. So that wobble in the codon actually is used here by the Sigma 70. And that's what I want you to get out of that. I might ask you a question on this paper. and what this paper is about. Horizontal gene transfer is what it's about, but it's actually the part that's technology that you want to know is the fact that, hey, I might need to adjust my codons, all right, to work uh, with a new organism when I'm taking a gene and putting it in there. So we know that there are multiple codons, all right, for a particular amino acid. And you think, well, it's worked in that one, it'll work in this one. It really doesn't matter what codons I use. But yes, it does. Because what you want to do is you want to get that sequence into the right GC percentage for the sigma 70 that is used in that organism. And you might also have to adjust things like the promoter. We've already seen that I've told you in our worksheet here that in our previous one that we want high and low GC and AT content at the promoter. Well, you need to go find out what this is for your organism. In other words, your organism, we're using K12. So we know it's roughly 51 and 49%, roughly 50-50. So anything above or below 50%. But there will be organisms, other bacteria, that this is not going to be the case. And because of that, you're going to have to adjust your analysis and you'll have to adjust your codons, etc. So I want you to know this. Maybe on a quiz, I'll eventually give another quiz. We haven't been doing them because you're working on this. But you will need to remember that these GC percentages, GC and AT, most people just talk about GC and it just implies that the AT is the other whatever's left over. So if I say 60% GC, that's also telling you it's 40% AT. So most of the time you just hear people talking about GC content, but they really mean GC and AT. It's just you can find the AT if you know the GC. So the idea here is that when we look at a organism, we have to look at percentage of GC and AT in order to get the genes copied efficiently, anyhow, by the sigma 70 protein that's part of the RNA polymerase holoenzyme. In other words, there are several proteins that make up the entire RNA polymerase, and some parts of them are sensitive to the GC content, and we have to adjust to that when we switch organisms. All right, that'll do it. The next few videos will be just on how to format your answers and how I want the project turned in.